Hi class, welcome back to the micro lab. Today we are going to be going over the gram stain and I will be demonstrating that for you. So we are going to go over the gram stain, what it is, why we do it. We're going to go over the steps first, so I'm going to go over each and every step and why each step is important, and then I'm going to demonstrate the gram stain for you. So starting out, we're going to talk a little bit about what it is. So the gram stain is a differential stain, and it was developed by a man named Christian Graham. So we've already went over the simple stain, and that's what they started doing in the beginning, was just staining all the bacteria they could get their hands on. Well, when they started staining the bacteria, they started realizing that some bacteria retained the dyes while others did not do as well of keeping the dyes. So they wanted to know why. Well, the reason why is because there are differences in the cell wall of bacteria. So all bacteria have a cell wall, but they look different depending on which bacteria you're dealing with. So pretty much all bacteria are split into two groups. They're either going to be a gram-positive organism or a gram-negative organism. So when you're dealing with a bacteria, it's really important for you to find out if it's a gram-positive organism or a gram-negative organism, because this is really going to help you identify what kind of bacteria you're dealing with if you have an unknown. So the gram stain is one actually that you would do that first before you would almost do anything else. Let's first talk about the differences in cell wall so that we know what we're looking at whenever we're talking about the gram stain. So, like I said, there are two main types of cell walls, a gram-positive and a gram-negative. So the difference is in the structure of the cell wall, so what the cell wall looks like and is built, how it's built. So the first difference is the thickness of the cell wall. So the main component of the cell wall is peptidoglycan. And in gram-positive cells, the peptidoglycan layer is very, very thick. So it has a really thick cell wall. In gram-negative cells, that layer is very thin. So in gram-negatives, the cell wall is very thin. So that's the very first main difference. Gram-positives have a thick peptidoglycan layer. Gram-negatives have a thin peptidoglycan layer. And of course, that's going to dictate whether the stain is going to stay or not. The second main difference is the presence of tessioic acid. In gram-positives, they have this acid that is running through their cell walls. It helps to strengthen the cell walls and make them stronger. Gram-negative cells do not have this. They have what is called an LPS layer, or a lipopolysaccharide layer. And this layer is on the outer part of the cell wall, and it's basically like a thin sugar layer. Okay, so in gram-positives, you've got a very, very thick peptidoglycan layer with the tessioic acid running through it. In gram-negatives, you have a thin cell wall with no tessioic acid. Instead, you have a thin layer of LPS lipopolysaccharides on the outer part of the cell. So you're going to need to remember that for whenever we start talking about the steps of the gram stain. So we're going to move on to talking about the steps of the gram stain. I'm going to go over each and every step, and I'm going to explain to you what's happening in the cells at each step. So like I said at the very beginning of the video, gram stains are what are known as differential stains. So you're going to see differences in the structure of the bacteria based on what color you see. So for differential stains, you see two different colors if they're done correctly. For a simple stain, all you're going to see is that one color, and you're able to look at the size, shape, and arrangement of the bacteria. For a gram stain, you can still see size, shape, and arrangement because you are staining the cell wall, but you're going to see two different colors. You're going to see a color for a gram-positive cell and another color for a gram-negative cell, and that's what you need to see is the two different colors. So starting out, we're going to make our smear, and you make your smear the same exact way that we did for the simple stain. You add your water, you add your bacteria, making sure that you're flaming in between, let it air dry, and heat fix it. So all those steps are going to be the same that you learned for the simple stain. The first step of the gram stain is known as the primary dye. So the primary dye, which is the first one, is crystal violet. Okay, so We flood the slide with crystal violet just like we did in the simple stain, let it sit for a minute, then rinse it off till the water runs clear. At that time, all cells are purple. The gram-positive cells are purple and the gram-negative cells are purple. So that's the primary dye. The second step of the gram stain is known as the mordant or the fixative. So just like the words say, you are going to be fixing something into the cell wall. You're going to be fixing the stain or the dye into the cell wall. The mordant in the gram stain is iodine. So you will apply the iodine, cover the entire slide, let it sit for a minute. So what's happening is the iodine is going into the cell walls and it's reacting with the crystal violet that's already there. It forms what is called a CVI complex. This complex is very, very large. So it helps to hold the crystal violet into the cell walls. So still at this point, everything is still purple. Gram-positive cells and gram-negative cells. Everything is still purple. The third step of the gram stain is the decolorizing step. So we are going to be taking the color out of something. Since gram-negative cells only have the thin LPS layer and a thin cell wall, as soon as you hit the cells with the decolorizer, the color is going to come out of the gram-negative cells. Now, the decolorizer in the gram stain is 95% alcohol. So we are going to rinse the smear with alcohol, and immediately the color is going to come out of the gram-negative cells. For the gram-positive cells, they have that nice thick cell wall, and that CVI complex is held tight in there, so the alcohol cannot take the color out of the gram-positive cells. So after the decolorizing step, you have your gram-positive cells should still be purple. Your gram-negative cells will be colorless because you've taken the color out of them. So the next step, which is the last step of the gram stain, is the counter stain, or the secondary stain is sometimes what it's called. This dye is safranin. It is a red color. So you're going to cover the smear or cover the slide in safranin, let it sit for a minute, and then rinse. At the end of this, your gram-positive cells should be purple. Your gram-negative cells should be pink/red because you've colored them with the safranin. Now, let's talk about how this can go wrong. If you leave the alcohol on too long, okay, it says in your lab manual 5 to 10 seconds, but you go even longer than that, okay, that can ruin your gram-positive cells also. So the color will come out of the gram-positive cells because even though they are pretty resilient to the alcohol, the longer you leave it on there, the more that alcohol can work. Eventually, that thick cell wall is going to disintegrate and you're going to have the color come out of that also. So you don't want that to happen. If you don't do the alcohol long enough, you get nervous, you only do it for a second, and then you stop, then everything's going to end up being purple. The gram-positive cells and the gram-negative cells. I always tell people that gram stains are kind of like an art. Even though you follow the directions, depending on the thickness of your smear, sometimes you get it right and sometimes you don't. That's why we always use controls whenever we're doing a gram stain. So our controls in this lab are always Staph and E. coli. Staph is a gram-positive cocci. E. coli is a gram-negative rod. We know what those are. So we put those on either side of the slide and we put our unknown in the center. That way we know for sure that we did the gram stain correctly if our staph ends up being purple and our E. coli ends up being pink/red. So that's what I'm going to do today is I'll have an E. coli smear and then a staph smear. I'm not going to do an unknown in the center because we don't really need to do that, but you do need to see what a gram-positive cell looks like and what a gram-negative cell looks like. So let's move over to the workbench. Alright, so we are over here at the workbench and I've already got my smears made. As remember I told you I'm not going to go over all the steps of making a smear. So I've got my smears made and I have staph on one side and E. coli on the other. So there are my smears and I've already heat fixed and everything. So I'm ready to move on to the actual gram staining step. So the first step, remember, is the primary dye, which is crystal violet. in the gram stain. And now there are other differential stains. The gram stain's not the only one, and they all have those 4 main steps: primary dye, mordant, decolorizer, and secondary dye or counter stain. So we're going to do other differential stains in here, and so they're going to also have those 4 main steps, except for the dyes themselves are going to change. So we're going to flood the slide with crystal violet, and we're going to let that sit for 1 minute. And the key here is that after each step, you remember to rinse. Okay, so after the primary dye, we're going to rinse. After the mordant, we're going to rinse. After the decolorizer, we're going to rinse. And after the saffron, we're going to rinse. It's really important for you to remember to rinse after the decolorizing step, because if you forget, that alcohol will be left on there and it will constantly be taking the color out of the gram-positive cells if you leave it on for too long. So you want to make sure that you remember to rinse in between each of these steps. Alright, so now we are going to move on to the mordant step. I've already rinsed my crystal violet off, and now I'm going to apply the mordant, which is iodine. And you treat it just like if it were a dye. You're going to flood the entire slide, and we are going to let it sit for a minute. Now, after this step, when you rinse, you don't want to rinse a whole lot, because you might potentially rinse some of the color out, and we don't want that to happen yet. We want that to happen for the decolorizing step. So we're going to leave this on for about a minute, and remember what's happening here. The iodine's going into the cell wall, forming that CVI complex, and helping keep that stain in the cell wall of the gram-positive cells. And like I said, we don't want to rinse too much. The first thing I'm going to do is kind of just tap this excess iodine off, and then we'll rinse it a little bit to try to get some of that iodine off. I only really rinse just to get enough of the iodine off to where I can see the smears, because you want to be able to see those smears so you can watch the runoff whenever you're doing the decolorizing step. Just a quick little rinse. I'm going to rinse the back off. And you'll see that that purple color is even darker now on that staph smear, because that's my gram-positive cell. Alright, so now comes the alcohol, and you're going to do this in a little different. You're not going to just you're not going to just put it on the slide and let it sit. You're actually going to want to see that runoff. You want to see it come off. And of course, my E. coli smear is the gram-negative, so that's the one the color should come out of first, and then the staph smear should stay the same. It should still stay purple. So you can already see that my staph smear is really, really dark, and then my E. coli smear is really, really light, okay? So it's kind of already hard to see. So we're going to rinse with our alcohol. That should be good enough. Now we're going to rinse. Okay, remember, you want to make sure that you rinse after the decolorizing step, because you don't want that alcohol to stay on there any longer. Now, if you look at it, you will see that the staph smear is still there, and my E. coli looks like it's completely gone. Don't freak out. It's still there. It's just colorless. So now we are going to counter stain with our saffronin, and that is going to stain our gram-negative cells. So again, you're going to let it sit for a minute. After the minute, we are going to rinse with water, and then we are going to blot with our bibulous paper, just like we did with our simple stain. And then we're going to look at it, and hopefully our gram-positive cells are purple, and then our gram-negative cells are pink/red. Your book says red, and a lot of places will say red, but I always say pink, because under the microscope it does look pink instead of a bright, bright red. So that's why I just use that term. Alright, so we're going to rinse the saffronin off. Alright, and we're going to get as much of that water off as we can, then we're going to blot dry. Again, being careful not to press too hard, because we don't want to crack our slide that we just worked so hard on. Alright, so now we've got our slide completely dry, And there is my slide. So you'll see that the E. coli is not as dark, very light because it's that pink color. And then we've got our purple smears for our staph. So now let's look and see what it looks like. Okay, so here is our gram-positive smear. So if you remember, we had two smears on one slide. We had our gram-positive cell, which was staph, and we had our gram-negative cells, which are E. coli. So this is what a gram-positive cell should look like under the microscope. Notice that nice, dark, deep purple color. And this is staph, staphylococcus, so it's forming those large clusters, and that's what you're seeing there. So we want that nice, dark, deep purple, and that's what we're getting. Now we move on to our gram-negative smear, and this is the E. coli, which is our gram-negative rod. As you can see, there's a really big difference between the color. That gram-positive was a nice dark deep purple, and this one we have a red/pink color. Like I said, the safranin dye itself is red, but under the microscope it looks pink. But that big main difference between the purple and pink, that's what you want to see. If you were doing an unknown and you saw this color, then that would mean that that is a gram-negative cell. If you saw the purple color, then that would be a gram-positive cell.